Top Med Talk.
Hello and welcome to Top Med Talk and our special series of podcasts, keeping you informed and up to date on developments during the coronavirus pandemic. I'm Joff Lacey and my special guest today is Professor Paul Heath. Paul is a consultant in Pediatric Infectious Diseases at St George's Hospital in London, with a specialist interest in immunology and vaccination. He co-leads the Pediatric Infectious Disease Research Group and is director of the Vaccine Institute based at St George's. Professor Heath, many thanks for joining the Top Med Talk team today.
Pleasure. Good morning.
Your team at the Vaccine Institute is at the front of this global race to produce a safe and effective vaccine for coronavirus. You have recently started phase 1 trials on a candidate vaccine. Now, can you give us an overview of this trial and what role your team at St George's is playing in the effort?
Yes, as you say, we are one of the sites in a phase 1, phase 2 vaccine trial with this vaccine. The vaccine is from Oxford University. It's called the ChAdOx vaccine, which means it's a chimpanzee adenovirus vectored vaccine. This is technology that is being developed at Oxford University. They have used this for other vaccine candidates, for example, most recently MERS, the Middle East Respiratory Syndrome, another coronavirus. And we're very excited to be part of this. The first phase is phase 1, phase 2, will be around 500 or so subjects and will proceed from there, we hope, if safety in particular is demonstrated, and we have every reason to believe that will be the case, into the phase 3 trial. And the phase 3 trial, of course, will be the big trial in which the efficacy of the vaccine against coronavirus disease, SARS coronavirus 2 disease, will be assessed. And we will be participating in that study, as will 4 or 5 other trial sites around the UK.
Which other centres are involved then in this first phase?
Oxford, Bristol, Imperial, Southampton, and ourselves. A group of investigators who collaborate frequently in vaccine trials. And we have a network called the UK Pediatric Vaccine Group, the UK PVG. And as I say, we often collaboratively work doing trials which need sizable numbers of subjects. So this is another example of that for us.
You said you're hoping to get 500 participants. Is that including all sites or just at the George's site? Or what's the numbers you're hoping to get?
The numbers at the St George's site will be around about 140, something of that sort, and it will be a similar number at the other sites. The final number is slightly in flux. That's a sort of ballpark. So it's going to be around 500 to perhaps 800 sort of subjects, something of that sort, and that will vary according to a whole range of things. There's a particular emphasis on trying to get healthcare workers vaccinated at this stage, and all of the sites are able to do a variable number of healthcare workers. And the obvious reason for this is that healthcare workers are likely to be at highest risk of disease, and therefore we would like to get them vaccinated as early as possible. The numbers recruited at each site will vary somewhat according to the number of healthcare workers who are eligible. And because this is a phase 1, phase 2 trial, the eligibility criteria are very rigorous. So the vaccinees have gone through a screening process which has involved bloods being taken to look at normal parameters, renal function, liver function, but a range of other things as well. And they will have been examined, and we will also have obtained the history from their general practitioners to ensure that the subjects are as healthy as possible, which is, of course, typical for a phase 1, phase 2 trial like this.
And I assume you also want to try and ensure that these volunteers haven't already had COVID and therefore already attain immunity. How are you going about that process?
Yeah, absolutely. The principal reason is based on history. So we are very carefully asking about whether they have been exposed to known cases of COVID-19. And then, if not confirmed cases, whether they have been exposed, for example, in the household, to individuals who have the features of COVID-19 but may not may not have had the test done. Now, in healthcare workers, particularly in frontline healthcare workers, those who are working on intensive care units and in the emergency departments, which is a smaller number, but potentially at a higher risk, we're also doing an antibody test. And this is a test that's been developed in Oxford. It's not fully validated, but it does provide, we think, a reasonable assessment of whether that person has previously been exposed. And if they have, then they won't be eligible for this phase 1, phase 2 component, because obviously we want to test the vaccine in those who are completely susceptible to disease.
Can I ask you a bit more about the composition of the vaccine? So you mentioned it's a adenovirus is kind of the vehicle for the vaccine. Is there a particular scientific rationale behind the design, or can you tell me a little bit more about that?
To be fair, the principal component with regard to the SARS coronavirus 2, the virus that causes COVID-19, is the spike protein. So the spike protein is a critical part of this virus, and we believe that neutralizing antibody against a spike protein will be protective. And so this vaccine is designed to express the spike protein and thereby, of course, elicit in the recipient immunity, antibody to the spike protein. And we hope, therefore, that will be the mechanism of protection. The way the spike protein is being expressed by the use of this vehicle, this vector, which is an adenovirus. And of course, adenoviruses are common viruses, and in humans, adenoviruses cause a range of usually mild common cold type conditions. And indeed, human adenoviruses have been used as vectors in vaccines previously. One of the issues with that is that because these are also common viruses, humans may well have pre-existing antibody, and that therefore interferes with the way these work as vaccine vectors. The solution that Oxford University team have gone for is use a different adenovirus, one that causes disease in chimpanzees. So it's extremely unlikely that in humans there will be pre-existing immunity to this particular vector, which is why it's been chosen. And as I say, it's been used in previous vaccine studies undertaken by the Oxford group, most recently with MERS. So over 300 subjects have been involved in vaccine studies with this particular vector. Although I should say that adenovirus vectored vaccines in general have been used in many thousands of subjects previously.
So if this adenovirus vector has been used successfully before, and you have this particular spike protein that you know is such a key component to the coronavirus activity, is there a pretty positive outlook for the success of this vaccine, given that structure and that previously known successful use of the vector before?
Yes, of course, is the answer, which is why it's progressing as rapidly as it is. But of course, the truth of this is that we do not know, hence the need for this series of studies, and in particular, the efficacy study, the phase 3 study, because we do not really know whether this will be an effective vaccine. There's every reason to believe, based on biological plausibility and the evidence, as I say, taken from previous forms of this vaccine against different viruses, that will be so. But I think we cannot say for certain. And indeed, when discussing with the volunteers for this vaccine study, of course, that is exactly what we need to discuss with them. And that's really important. But this is there is no licensed chimpanzee adenovirus vectored vaccine that we can look to say that this technology is effective at preventing disease. So that's why we need to do this big trial. And I think, to be fair, that's true of all COVID-19 vaccines at this moment in time. Clearly, the global pandemic is unprecedented, and the vaccines that we need to prevent disease are also untested and unprecedented, using technologies that haven't been used before and certainly haven't been used in licensed vaccines. So this whole area is, to a large extent, novel. But of course, it goes without saying that there has been incredibly careful thought about whether this technology is, first of all, safe, and then, as I say, effective, based on all the principles that we have And we have learnt from developing and testing vaccines over many years.
Can you now explain exactly how this trial will play out? What's the design?
The phase 3 trial is a double-blind, randomized controlled trial, so a classic phase 3 trial with the endpoint being disease itself. And so participants will be randomized to 2 vaccines, one being the ChAdOx SARS coronavirus 2 vaccine, and the other, the control vaccine. Now, the control vaccine is a meningococcal vaccine, so against meningococcal disease. It's a quadrivalent meningococcal conjugate vaccine. And that's been chosen because we know that vaccine to be safe and indeed effective. And also, it's unlikely that many of the recipients will have received it before because it's part of the adolescent program now, only recently. So many of the subjects in the relevant age group won't have received it. And even if they had received it before, we also know that receiving 2 or indeed 3 or more doses will not cause any safety issues. But the importance of having a control vaccine, of course, is that we can ensure that the recipient and the person vaccinating and assessing the response isn't aware which vaccine they have received, and therefore their assessment of their responses is completely unbiased. And that's a critical component of a phase 3 trial like this.
Can I ask why use an actual vaccine as the control? Why not just give saline?
Yeah, well, that's a very good question. And of course, that would be a possibility and has been used before in such trials. There's no right answer to this because a saline placebo would also be appropriate. But a saline placebo won't offer any potential benefit at all, of course, to the recipient. A vaccine effective against 4 different types of meningococcus has the potential to be effective. Now, the risk of that individual going on and being exposed to any one of those meningococcal strains is, of course, very small. But as I say, there is the potential that it could prove effective for that individual as well. So there is potential benefit there, albeit small. And I guess also it's true to say that this is the vaccine that has been chosen as the control vaccine is one that we're very familiar with. So we know what its safety record is. We know what the reactogenicity will be when administered to subjects. That is the sort of rates of redness and swelling and fever and so on. So we're very familiar with this control vaccine. But it would also be entirely possible to have chosen a saline placebo. I think increasingly many groups will be using for their control a vaccine that has some potential benefit. But that, as I say, is something that might vary in different studies.
Got it. From the point of vaccination, what's the follow-up program for the volunteers?
Well, it's very careful and very detailed because we're thinking about 2 things here. One is safety and the other is efficacy. So we need to be able to capture any episodes of, in this case, COVID-19. This is part of the discussion, of course, that happens with the participants prior to and after vaccination. And they will also be prompted on a regular basis for symptoms and signs of possible COVID-19. And if they develop any of those symptoms and signs, then they will be instructed to make contact with the study team. And then a sample will be taken, a swab will be taken to see if, in fact, they have got COVID-19. And then, in fact, that's repeated again some 7 days later. By this way, we hope that we will capture all of the cases that occur during the course of the study. But of course, we need the participants to buy into that. But that's all part of that process before they are vaccinated, the screening process, the informed consent process, because it is critical. And equally, as I say, is the safety issue. And the safety issue is all about telling the subjects that they need to make contact with us if indeed anything is of concern for them. But there's a clear list of particular items that would be of concern, some of which because they will potentially be related to the endpoint of the study, COVID-19, but others which could conceivably happen in the context of vaccination. So very rare neurological events that might happen Guillain-Barré syndrome, for example, has been recorded in vaccine studies in the past. So we obviously ensure that we will hear about and will assess any potential side effect and any potential endpoint disease in the recipients during the course of the study by very detailed and close follow-up.
The trial relies on people in the study contracting coronavirus, COVID-19. How long do you think it will be before enough people have been affected that you're then able to ascertain if the vaccine has been effective?
Yeah, I mean, that's a really good question. And it's a major issue, of course, in assessing the efficacy of the vaccine. We need the cases of disease to occur. And I think it's true to say that to a large extent, the previous coronavirus vaccines or vaccines against the more severe coronaviruses, SARS coronavirus and MERS coronavirus, have stopped in progress because disease has to an extent disappeared. And so it has not been possible to assess the efficacy of those candidate vaccines. And of course, we're seeing that here now with COVID-19. So we see that in the UK, the numbers of cases has plateaued. And certainly in certain parts of the UK, and there is a regional difference, it would seem, is going down. So the opportunity to assess the efficacy of the vaccine is declining as time goes on. Now, of course, there is the possibility of later peaks of disease, and we do not know how this disease is going to behave, whether it comes in cycles, whether we will see it again in the winter months, for example. So I think the answer is that this was a major reason for accelerating the progress of this particular vaccine into clinical trials. And indeed, I think it's a major reason for being as flexible as possible during the course of the trial. And it may be, for example, and I don't know that at all, but the sorts of things that the investigators now will look for is, well, should there be sites in different places that have not yet reached their peak of disease? Are the subjects in the study at highest risk as possible? And obviously one of those groups is healthcare workers. But as time goes on, it may be that there are other high-risk groups that could be targeted for this trial. And for example, teachers, perhaps, if the schools go back. I don't know that. The point is an incredible amount of flexibility, I think, is required, and a constant view of what's happening in the population with regard to the disease to ensure that this vaccine is given the very best opportunity to show whether it is effective or not. And that's so important because we all need, of course, an effective vaccine, a vaccine that can then be rolled out through our population, the UK population, but of course, globally as well. And so this is a really important stage for vaccine development. And this is the case, of course, with other vaccines that are being developed, although very few of them have so far reached human trials, but many of them are coming to that point. And all of the investigators, I think, it's true to say, are concerned about the fact that they will miss the opportunity to test the efficacy of their vaccine if the disease is going away, which, of course, in many other respects is a great thing. So we have this difficult philosophical aspect to this. We want the disease to go away, but we need the disease
you want to test the vaccine. Yeah,
indeed, yeah.
So this may be an impossible question to answer at this stage, then, but is there a possible or projected timeline as to when, if all is successful in terms of determining efficacy and safety, a timeline as to when you would see the widespread use of a vaccine, of this vaccine?
Well, really hard to answer that. But what I would say is that it's clear that our previous figures and understanding of how long it takes a vaccine from development to implementation has been accelerated hugely as a result of all of this work in the context of the pandemic. So all of our previous assumptions go out of the window because normally it takes years, of course, from a vaccine from start to finish, years. That won't be the case here.
Out of interest, so you've got to the first phase of a human trial in a seemingly very short period of time. How different is the timescale of this current project compared to more regular vaccine development projects? How quickly have things been accelerated?
Hugely. I mean, to be fair, the first vaccine against COVID-19 to go into humans was the Moderna vaccine, which is an mRNA vaccine in the United States, and that went into humans in March. So that is unprecedented. I mean, this trial is unprecedented as well. We normally think about years, 10, 12 years more. From it depends on the vaccine, of course, but it's that order, it's that sort of ballpark. Here, we've gone within months into human trials. And then, as I say, the expectation is, well, what we were saying a month ago, I think, is that, well, maybe 12 to 18 months before implementation. And I guess that sort of figure still holds, although I have heard more confident people talk about the end of this year, into rolling out potentially to high risk groups such as healthcare workers, a vaccine such as this one. Clearly we don't know, because we don't know what's going to happen next. We don't know whether there will be safety concerns with this vaccine. We don't think there will be. We certainly don't know whether this vaccine will be effective. And that clearly is the question. And we'll know that later this year. What is also happening, as far as I'm aware, is that mass production of this vaccine is already being planned and perhaps underway, such that it will move in the mass production, the mass manufacture of this vaccine will move in parallel with this phase 3 trial. And clearly that never happens normally, because that will wait until the trial has gone through and come back with results. But that is not happening here. And I think that example is also true of many other vaccines that are moving through this process, because people are willing to spend the money to commit to getting a vaccine that can go straight into implementation when all of those other points have been satisfied, which means, of course, they are potentially there will be considerable loss of money if that bet is wrong. But I think that just demonstrates where we are at the moment with this global pandemic and just this overwhelming urgent need to have a vaccine for a huge number of people. And that has meant that, the normal assumptions are no longer relevant. We're in a completely different place to any other vaccine that we have worked with.
Absolutely. And a final question I just wanted to ask, do you anticipate that a successful vaccine would provide lifelong immunity for the coronavirus, or is this the kind of a structure that would need a repeated or booster project to maintain immunity?
I mean, another great question, and I'm afraid another great question to which I don't have a great answer. I mean, time will tell. Time will tell. We do not know the answer to that, but clearly on the back of this study, this trial, will be the need to look at the persistence of immunity. Indeed, already as part of this trial is a 6-month follow-up, and that's the efficacy follow-up, but also a 12-month follow-up, 12-month blood test to look at persistence of immunity. And that undoubtedly will be followed by longer follow-up to see how long immunity antibody will last. We need to know more about what antibody means. We need to know whether what we're measuring equates to protection. That will come with time. Part of that will be built into this trial as well, in that antibody levels will be taken in all of the recipients, and then whether they go on and develop disease or not can be related to the amount of antibody they did or did not have. And so trying to devise what might be called a serological correlate of protection is really important, because then that can be used to see how long that level of immunity, that level of protection might last. And it may well be, as with other vaccines that we use, booster doses will be required at variable periods of time afterwards. But we don't know the answer to that question. And that, as I say, will come from these studies, as well as from really good surveillance for disease. And it's from enhanced surveillance of disease that we also start to see whether booster doses might be required. And that's how we come to 2 doses of MMR, for example, and for other vaccines that we have. We've learnt from very close monitoring of disease, surveillance of disease, when additional doses might be required. So it'll be from a combination of those types of studies that question will be answered. So I'm afraid only time will tell.
Well, Paul, we must leave it there, but thank you very much for your time today, but also for your work on such a critical project in our effort to fight this pandemic. And I wish you and your team all the best and success with the trial.
Top Med Talk.
Thank you for listening to Top Med Talk. You can catch the remainder of our podcasts on the website. But for now, thanks for listening.
Top Med Talk is proud to thank its sponsors, specifically Edwards Life Sciences, for their generous support through this global crisis.